forget. There we go. So 7th period, this is Thursday, November 12th, and this is going to be your first day of lactase data collection. But before I go there, I want to make sure that that we just do a little bit more of that pear deck that I showed you, which I think is going to be really helpful. So I'm just going to share that pear deck with you just a second here, just to finish up a little bit of, I think for the most part, you guys understand, you guys have enzymes under your belt, but there's a couple things. Okay, let's start yeah, I'm gonna have to do a different screen. Okay, all right. There's a couple things that about enzymes that you may have forgotten from your freshman year. I'm just gonna go over a few things before I then show you this really cool test tube simulation that will help you collect data for this lactase lab that we're starting today. You're going to start this lab today, and it's going to take us, 7th period, it's going to take you a longer time to get it finished. You'll do the last part of it. You'll do the last part of it after you take your unit test. Your unit test is still Thursday next week. Right, your unit test is still Thursday the 19th. So make sure you mark your calendar for that, because we're still going to be ready for that. But you guys won't finish your lactase enzyme group thing until that one day that we have together, Thanksgiving week, is when you guys will do that. And if you guys have scheduling issues, don't worry, we'll fix it. We'll get you in another time. Don't worry, okay? But there's a lot of data collection and data prep work that needs to happen that you're going to need help with before then. So today is finish talking about enzymes, let you collect some data using this simulation, which doesn't take a lot of time at all, and that you'll be able to do right here. And then next Tuesday when we're together will be a catch-up day for you to finish the graphs if you've had trouble with them, figure all that out. Ask me last-minute questions about enzymes, and then your final group part of it won't be until after the test. Okay. But first of all, yeah, let's just go into this pear deck and just talk about enzymes for a few minutes. So I'm just going to wait for you guys to get in here. And just like you did last time, if you want to write things down, fine. But remember that this will be posted for you. This presentation will be posted, so you can always go back into it and get stuff later. So maybe you just want to just participate today, and you can put notes in about enzymes, excuse me, at a later time. Okay. I like the frog. Cute. Okay, so if you want to join after the fact, that's fine. The joining code will still be in there, but I'm going to go ahead and start the pear deck. Okay? Okay, so we got through a lot. I'm just going to go all past this because we already did all this. We talked about entropy and exergonic and endogonic, all that. We did that. So I want to start. Oh, wait, there it was. Right there. Okay. I like this picture because this is a really nice sort of summary picture of what the enzyme is and what it does. So the big blue thing is an enzyme, right? And this is the enzyme before it connects to its substrate, which is this little red thing here. So when the substrate connects with the enzyme, it connects in a place called the active site. And the active site is this perfect little pocket in this large protein. Remember, this is a protein. And the substrate fits right in there, and that becomes kind of a perfect little microchemical habitat for the substrate to either be pulled apart, sometimes that's what the enzyme is doing, or sometimes there's two substrates that go in there and they're perfectly oriented so that they will be put together. The enzyme provides an environment for a reaction to happen that is a much more efficient environment than if the substrate was just floating around in solution by itself and trying to react spontaneously by knocking into other things. So an enzyme speeds up a chemical reaction very, very, very much. And that's important for living things, because if these reactions, like digestion reactions or synthesis reactions, if they happened really slowly, which they would without enzymes, because it's just not efficient for them to react, you wouldn't be able to fuel all the metabolic needs of a big, expensive organism like a human. So enzymes are really lifesavers. And again, one of the reasons why doctors get nervous when you get a high fever is because when you get a really, really high fever, the enzymes in your body are used to, 98.6 degrees. If it goes up to 106, 107, the shape of the enzyme can kind of unravel. And if the shape of this active site is the wrong shape, that kaputs your enzymes, and your enzymes are making everything in your body work. So that's why, for a human, that's very dangerous. Okay, so let's go on to this. So how does this happen? Inside the active site, you lower the activation energy barrier by some of the things I was just talking about. You orient substrates correctly, right? So if the enzyme is specifically supposed to take like 2 glucoses and make a maltose, then its active site can accommodate those 2 glucoses and put them right in the right spot for that new glycosidic linkage to form. A lot more likely that that's going to happen in an active site than if they're just bumping around randomly on their own. That's one of the most important things that the enzyme does. The other thing it does, which you're going to see in the animation I'll show you in a few minutes, is that if there's some basic R groups sticking out in there, if there's some ionic R groups, they can sometimes pull on a substrate and help its bonds to break. Okay, so I'm going to go past that. I don't want you to worry about that. Let's just keep going. I know you've seen this picture before. This is kind of from freshman year. And you can see that the purple thing is the enzyme, and that substrates are these red and green things, and they go into the active site. They are changed in some way, rearranged, their atoms rearranged, and then they leave and become the products. And then the enzyme goes back to its original shape. Enzymes are not consumed by reactions. They go back and they're ready to receive more substrates. Okay, so I think you can take a minute to answer this, because this feeds into our lab really well. Well, what kind of environmental factors do you think could affect whether an enzyme is going to work well or not? What do you think of that? Okay, oh, you guys are giving good responses. Let's see. That's it, pH and temperature, you've got it. Yes, those are the 2 big ones. And those are actually the ones we're going to be testing in our lab. Substrate concentration, excellent, that's also true. If you did the guided reading, you would definitely know that, so good for you, yes. pH and temperature are the big ones. Somebody else also, yes, salinity, nice job. So that's good, that's also another one. So thank you for paying attention and participating, you guys are awesome. Okay, let's keep going. Yeah, so see, temperature, pH, substrate concentration. There's also some specific chemicals that can act as inhibitors or cofactors. We're not going to talk a lot about that. Does somebody have any question? Is time... I think the only way that we really incorporate time into these stories, Carter, is that when we're looking at how fast an enzyme can turn a bunch of substrates into products. There was a lab that we used to do that we couldn't do this year, where we actually left an enzyme in contact with its substrate for different amounts of time. So that's as close maybe as you could get, where we would put like a potato into some hydrogen peroxide and it would react. But then if you squirted in some sulfuric acid, which denatures the enzyme, you squirt it in to stop it at different points. So the enzyme is in contact with the substrate for different amounts of time. But really, you don't need to worry about that. In terms of environmental factors, time is sort of unlimited, right? There's no, the cell isn't setting a timer and then squirting sulfuric acid at its enzymes. So in the natural environment, not really. Good, great question. Okay. Okay. Effects of temperature and pH. Here we go. So what you're going to see is that each enzyme has an optimal temperature in which it can function. Optimal means best, right? And this is also true for pH. There's certain pHs for each enzyme. So different enzymes have different optimal temperatures and pHs depending on where they have evolved to function. So there are actually enzymes that can survive very, very hot environments. There are living bacteria that live in like the boiling springs in Yosemite. If you've ever been to Yosemite, there's some boiling springs there because of the volcanic action. And the enzymes in those bacteria living in the springs can actually survive that environment. That's very rare though. Most enzymes cannot do that. If we put ourselves in the boiling spring, we wouldn't last very long, right? But it's the same with pH, it's the same with temperature. Each enzyme has its own different optimal temperature or pH depending on where you find them. Okay, so here, try your luck at this. Draw on this a curve that you think would correctly show the activity of an enzyme at increasing temperatures, and we're talking about salivary amylase, which is in the human mouth. An ideal body temperature, not in Fahrenheit, be careful, not in Fahrenheit, this is Celsius. So your ideal body temperature, 98.6, converts to 36 degrees, 36 or 37 degrees Celsius. So what kind of curve do you think you would have here for temperature effect on this human enzyme? Let's see how are you doing on this. Yeah, that's good, actually. That's just right, is that it would peak right at 36 if you did something like that. Yeah, what you get are these sine curves is what you get. Yes, that's exactly what happens. That you get curves that have peaks at around 36. And if you go too high, very, very high temperatures will denature most enzymes. And if you go too low, the enzymes won't work very well because they'll kind of slow down, right? So yeah, you've all got that. Very good. Yes, and the smiley faces. Thank you for that. Okay. All right. Let's go on to the next one. All right. Now let's try pH. Remember, pH is on a scale of 1 to 14, which with 7 being perfectly neutral. Going downwards towards 1 is more and more acidic. Going upwards towards 14 is more and more basic. So in the human stomach, we're talking about an enzyme that's adapted to work in the fluids of the human stomach. So where do you think pepsin is going to peak is the question. Take a guess at that. Okay. Okay, let's see what you thought. All right, let's take a look. Yeah, that's about right. It would peak in the acidic area, right down. And it really would, instead of a straight line, there is actually more of a sine curve with the peak around, believe it or not, the peak is around 2. Can you believe the stomach is that acidic? Hi, Charlie. This is my son, Charlie, sitting in here. Hello. This is AP Biology. This is my son, Charlie. George Mason student, music and jazz. We're very proud. Okay. So yeah, here we go. Yeah, that's about right. Yeah, it would be a sine curve with a peak. Believe it or not, it's about 2. Yes. And anything above or below that, there doesn't tend to be a tremendous amount of activity for pepsin because pepsin is used to working in a very acidic environment of the stomach. And more smiley faces. Yeah, that's good. Okay. All right. So let me show you. I think there's this next picture there. See there? Okay, so temperature. Here are actual temperature curves for 2 different enzymes. So like your salivary amylase peaks right around 36, 37, right there, right? So your sine curves there show the optimal temperature for a human enzyme is about 37, right? And then for thermophilic, like that bacteria that live in Yosemite, right? They can survive up to about 77 degrees Celsius before they really peter out and denature. Okay, and here's the pH one. So look at that. It's less than 2. That's really acidic. Did you know your stomach was that acidic? That's kind of interesting, isn't it? You've got a little vat of acid inside you that not only helps break down your food, but it also protects you against invading bacteria, because a lot of bacteria don't like all that acid either. So it protects you. And then the optimal, look at trypsin. So in the intestine, this is a different place from the stomach. After your food and materials leave the stomach and they start traveling through the small intestine, where a lot of digestion reactions occur, it's actually slightly basic in there. Look at that. So it's a little bit basic in the intestine. In the bloodstream, it's right about here. 7.4 is actually your blood plasma's exact pH, and it literally has to stay on that point. If it goes above or below it, you can be very, very sick. Okay, so that's where we're going to stop. We're not going to do any more pear decking, right? Okay, here I am back. So now I want to show you an animation that I found that pulls everything about what we've learned about enzymes together. It's a really good one. I already uploaded it so you can look at it yourself if you would like. Yes, where is it? Here it is, how enzymes work. Yep. And of course, okay. So I'm going to share this with you because I think this will kind of pull everything together. As far as all those energy graphs we looked at. Here we go. Okay, I'm going to turn off my sound before I go there so you can hear it really clearly. Just listen to this. Okay, you can hear me right now, right? Yes? Okay, all right. I don't know what to do other than to leave my own sound on and see if that helps, okay? Okay, hold on a second, I'll rewind it. That's weird. Okay, my sound is on. Let's see if it works this time, okay?
Every single second, inside every living cell, thousands of chemical reactions are taking place.
Is that better? Yes? Oh, good. Okay, I'm going to keep going then. Good.
These reactions are performed by enzymes. An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which is a series of chemical reactions used by cells to generate energy from carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. The life-sustaining power of enzymes lies in the fact that they catalyze reactions at mild conditions of pH, temperature, and atmospheric pressure. The rates at which allows reactions are modes to trillions times faster than those of the same reactions on biomass.
Okay, so this is a nice picture to freeze it on. Let me just kind of talk about this for a second. Those blue-green, spongy-looking things, those are the enzymes, okay? And they're much bigger. Usually the enzymes are much bigger than the substrates they're working on. So let me identify the things that are in this picture. The blue-green thing is an enzyme, fumarase, to be specific. And fumarase works on this small carbohydrate, which I think there's one right here. And there's one right there. There's the substrate right there. The rest of these other molecules, these are water molecules here. There's lots of water molecules around. And then there's the substrate, and here's the enzyme. So this is the catalyzed reaction, which, as I explained before, goes much faster than the uncatalyzed reaction. This is just a sugar molecule there, substrate, just bumping around trying to react haphazardly with a lot of water molecules around it and no enzyme to help it. So again, very much faster when there's this blue-green enzyme there with a nice active site to help it along than when it's just bumping around trying to react at random.
To speed up a reaction, the acids additional energy needed is heat, which jostles the substrates and occasionally requires reaction. In the course of most reactions, an unstable transition state is formed as the substrates are transformed into rocks. This is a time for the reaction. Into the substrate and putting it in the proper position to
Did you see that? That's absolutely perfect. And I love it because it even takes you to the energy curve at the same time. So there's this big orange thing here that looks like something out of Coraline, right? This is the enzyme, and this is the active site in here. And the active site is just the right shape to receive the reactants or substrates, right? So they go in and they get themselves inside the active site, and they're oriented just right to react together. And then you'll also notice that the curve reaching for that transition site is much lower, right? It takes a lot less energy when there's an enzyme there, and then it goes down. So let me show it to you again. Oh, okay. Now look at this curve and chat to me, because I know this isn't. Hold on. Is this curve exergonic or endergonic? Can you chat me an answer on that? Is this an exergonic or endergonic curve here? Look where the reactants were, where the products are. Oh, you guys are so great. Yes, exergonic, that's it. Yes. Okay, because the reactants, that's my dog. The reactants have more energy stored in them than the products. So there's a negative delta G here, and that means that energy must have been lost to the environment somehow, maybe by heat, or maybe it was stored in a new molecule. So very good if you got that right. Let's keep going to the end. I need help, Charlie, please.
To understand how enzymes work, let's take a closer look at the reaction in citric acid cycle that is catalyzed by the enzyme aromatase. A aromatase binds to the substrate citrate, moves a hydroxyl group and the hydrogen to form an intermediate cis-plant. It then adds the hydrogen and the carbon back and moves them in different positions to form the aromatase citrate. Some amino acids are perfectly positioned to recognize the substrate.
Okay, so here's a wild picture here. Let's explain this. This is the active site in here. All this gray twirly stuff out here is the rest of the protein, the rest of the enzyme, right? A long polypeptide chain that's out there, right? The black and red parts here are the R groups sticking out from amino acids that are right in that active site there. This R group has some iron and some sulfate in it. Sometimes enzymes have those things in them as functional groups. Here's another R group coming out over here and another R group here from the amino acids coming out from the, that interactive site, right? The molecule they're working on is right here in red and black. The red and black sugar is there. And watch as the different R groups kind of gently pull it apart and put it back together.
first step in the reaction is dehydration. The next step is to deacids and acid proteins that rise on the substrate. Serine then acts as a base, exciting the opposite side of the substrate, forming an cis-alone. Cis-alone then flips upside down and the complementary combination. In this step, histidine grabs a hydrogen atom from the acid compound, placing the resulting hydrogen back onto the substrate. Serine then returns its hydrogen atom, and the final product process is mixed. Notice that the enzyme itself was unchanged by the chain, and it's not a hydrogen group and a hydrogen atom, thus starting and ending the same step. This is the hallmark of the catalyst. When it flips its reactions, it is a mix, resulting from thousands of reactions in a row. Notice also that the shape of the active site is flexible, and the enzyme surrounding substrates, closing them to form the proof environment of reaction.
Okay, that's actually the end of that video. So what did you think of that? You can still hear me, right? Yes. Okay, so obviously you don't have to know every detail of what that of the R groups and the serines and all the things he was talking about, but that gives you a really good idea of what goes on in an active site, right? The substrate went in there and then it was gently pulled apart and stuff was replaced by the inner molecular environment of that active site because of those particular amino acids that were lining the active site and the R group sticking off them, that particular molecule was able to go through its changes. So you see how very specific enzyme active sites are. One enzyme can only work on one particular substrate, and it does that thing and then sends the substrate on, sends the product on to maybe the next step, okay? So that is all I had to tell you about enzymes. Now I'm going to stop this recording and start a new one where we're going to talk about the lab, where you're going to play with a little simulation. But before we do that, I think we should take a little break, all right?